Hello, and welcome to the Mushroom Mindset, Enhancing Cognitive Function with Ergothionine. This is the first event in a series of on-demand webinars that I'll be recording for Real Mushrooms. And I'm Lee Carroll, Chief Medical Herbalist. Before we get started, I need to just read this disclaimer briefly. I'll get you to pause the video so you can go through the whole thing. I won't take up the valuable time here and read it verbatim, but you do need to know that this is information is for healthcare practitioners only. It's for their educational purposes, where we haven't evaluated the legal status or the FDA status of any of the things that we've talked about. We're not offering medical advice. We're not offering to cure or treat any particular disease. And as healthcare professionals, you do need to do your own due diligence. So in this brief presentation, and I'll try and keep these events under an hour, preferably about 45 minutes. Some topics just deserve a little bit longer. So in today's session, I'm going to introduce you to what ergothionine is, the basics of how it works, but not get too kind of into the weeds in that regard. Just give you an appreciation of just, what it does and how important it is. So you've got a framework upon which to, hang your discussions when you talk to patients and when you think about it in terms of how to use it in particular clinical settings. So ergothionine is important across a whole range of health areas. So I'm just starting off with cognitive function because there's really good research there and it allows the story to unfold in a really natural way. We'll talk a little bit about safety and finish up with what my dosage recommendations are for ergothionine in general health and also across the board in terms of the different aspects of cognitive function. So for many of us, when I say the words mushroom and cognition together in the one sentence, we automatically think of lion's mane. However, many of us are just not aware that any edible mushroom consumption, so think agaricus, like portobello or crimini or button mushrooms, shiitake, oyster mushrooms, enoki, any of these mushrooms are also associated with significant improvements of cognitive function and prevention of cognitive decline in aging populations. And the research is really quite compelling in this regard. So there's at least 10 studies, and I'm quoting them all here. The references are on the next slide. So the total number of people in all of these studies is close to 60,000, and the data is quite impressive. And what the data says is that eating mushrooms, any mushrooms, once or twice a week is associated with significant improvements in how your brain works, particularly the cognitive part of your brain, across all of the domains. And it also helps to prevent cognitive decline and dementia in aging populations. So in younger people and those that are aging, it allows you to maintain your cognitive function or even improve it if it's a little bit off its game. But if you're dipping into mild cognitive impairment or dementia, it can actually help. So it doesn't matter what mushrooms you eat. And the most recent study here, you can look at the reference on the next slide, was published by a researcher called Char. They followed 30,000 people from the UK for 18 years, and they showed that just one portion of mushrooms, fresh mushrooms per week, was associated with improved cognitive scores across 7 different types of cognitive tests, including the mini mental state exam, which is very commonly and often quoted. If you bump that number up, in a different study, 13,000 people were followed for nearly 6 years. These people were 66 years old, and 2 or more servings per week was associated with a 57% reduction in the incidence of mild cognitive impairment. And there's an inverse relationship. So the more you eat, the less risk there is of developing dementia. These types of serves of mushrooms are also associated with reduced rates of depression and metabolic syndrome. And these are conditions in their own right, clearly, but they're also known risk factors for the development of cognitive decline. And You could add non-alcoholic fatty liver disease in here as well. So here's the references if you're curious to go in and have a look. So following on from that, I guess the big question is, well, how can dietary consumption of such low quantities of mushrooms have these significant associations? So they're not causation, but there's clearly something going on. So what is it about mushrooms? What are the constituents? What could possibly be the mechanisms of action that would plausibly explain this? And if we look at the chemistry of a mushroom, a number of candidates do stand out, and they are, the obvious one is beta-glucans. We've got ergothionine is another possibility, and the next possibility would be secondary metabolites. So it's very unlikely to be the secondary metabolites. If we use a lion's mane, for example, lion's mane's got haricinones. Lion's mane mycelium's got erinacine A, which seems to be attracting a lot of attention. But those low doses of mushroom don't deliver significant quantities of those secondary metabolites to have these effects. And people don't eat the mycelium. So, in terms of the diet, that cannot play a role. It's not even possible. The beta-glucans, from all sources really, when you look at the research, beta-glucans get fermented in the gut to short-chain fatty acids, and those various short-chain fatty acids are linked to enhanced temporal order recognition memory. They're linked to increases in brain-derived neurotrophic factor. There's a number of other significant impacts as well, like the microglia in the brain, so the glial cells in the brain are less inflammatory, and the brain just clearly works better under the influence of this gut-brain axis. However, just like the secondary metabolite argument, there's not enough beta-glucans in those 1 or 2 serves of mushrooms a week to generate these kinds of effects. You need to have much higher doses. So, the idea that any mushroom can support brain health is a separate discussion. And then if we get the dose right, the beta-glucans certainly play a role. So that leaves ergothionine. And I'd like to say that it's the most important antioxidant that the least number of people know about. And the level of ergothionine in 1 to 2 serves of mushrooms per week is enough to generate these effects. The human need for ergothionine is quite low, and 2 serves of mushrooms a week is enough to give that to someone. And there's very, very clear and plausible mechanisms of action that could drive these effects. So this then begins our journey into what ergothionine is. It's an unusual water-soluble molecule. It's got sulfur attached to it. That's the yellow atom spinning around on that molecular diagram there. And it's a modified amino acid. 2 amino acids come together to form this, but technically it's a derivative of histidine. It's in a chemical class that we call thiols. So it's in the same class of glutathione, which is the most well-known of all of the thiones. However, unlike glutathione, ergo, and I'll probably refer to it as ergo as we go along because ergothione's a bit of a mouthful. It possesses this really rare structure, and I'm not a chemist, so I can't wax lyrical about the uniqueness, but scientists say that it's a unique structure, and there are no other human endogenous metabolites that share this structure. It's totally unique. And just like a vitamin or an essential nutrient, humans need to consume it, and ergo as part of our diet is essential for optimal health and our physiological requirements. And I'll build the argument, with regard to that as we go through the presentation. So it's a potent antioxidant, and probably a better word, technically it's an antioxidant, but its main kind of action in the cells is cytoprotective. Its job is to play the role of supporting the cell using its antioxidant and to a lesser extent its and metal chelating properties, but I'll go through the list as we get through the presentation. So in fungi, and there's a very tight link between humans and fungi. If you go back in the evolutionary, kind of tree, fungi and animals share a common ancestor. So as a result of that shared common ancestor, there's about 30% of the DNA which we share between fungi and the animal world. So, we see that with ergosterol and cholesterol, and it's a very clear example with ergothionine. So in fungi, ergo performs the role of the principal intracellular antioxidant, and glutathione plays a minor role. Mushrooms, fungi in general, also have a thing called mycothiol, which plays a antioxidant protective role as well. But in humans, it's the other way around. Glutathione is the primary intracellular antioxidant, and ergo is a buffer, playing a secondary or a backup role. So we have to see ergo and glutathione as a team, and they work very, very closely together. And just like a tag team in wrestling, and when I was a kid, I used to watch the tag team wrestling, and as one wrestler is flagging, they reach out and they tag the other wrestler, and they can come in and help out. So that's what it's like with glutathione. As its levels are waxing in the cell as a result of oxidative stress, it needs to be regenerated. And in the time it takes to regenerate, ergothionine gets tagged to say, hey, you take over and support out the cell while I'm in a state of regeneration. So ergo was first discovered in 1909, and it came from the Claviceps purpurea, the ergot fungus, hence the name Ergothionine or thionine. So that's where its name comes from. And it's found in all plants and animals, even though they don't produce it. Plants rely on symbiotic fungi and bacteria in the soil to get their supply, and the animals need to consume other organisms that contain it, like fungi or plants, or if you're a carnivore, you might need to consume another animal and eat their liver or, eat their blood, for example. So humans have got a dedicated transport system that's in the cell membranes to bring ergothionine in. So when we consume ergothionine in our small intestine and our large intestine, these transporters, and they're uniporters, they just move the ergothionine in one direction. So they bring it in to the bloodstream, and then when it's in the bloodstream, those same transporters exist in many, many cells in the body, and then the ergothionine gets brought into the body, into the cells. And then it can't leave the cells unless it's metabolized, because that transporter is just a one-way doorway. So humans accumulate it, and we avidly hang on to it, and the half-life is about 1 month. So if you took 10 milligrams in a month, you'd still have 5 milligrams in your body. So if you take 10 milligrams every day, you can clearly build it up. And it's non-toxic, as we'll see later on. And it's clearly biologically important. Why would we have unique transporters to bring pretty much just only that one molecule if it wasn't important? Why would we have this cellular machinery? Why would there be this natural interaction between glutathione and ergothionine and all of the other antioxidant members if it wasn't clearly part of the the evolutionary kind of design or plan, or however you want to think about it? Many people are now calling it a vitamin, and Professor Bruce Ames was the first one, I think, to propose this theory, and vitamin L clearly stands for longevity. So proving that this substance is a vitamin is actually quite a challenging thing to do. And classical vitamins, like vitamin A and B1, B2, B3, vitamin C and D, they were all discovered because inadequacy of them leads to deficiency diseases like blindness and beriberi, scurvy, rickets, and so on. So proving that in humans with ergothionine is actually quite difficult to prove, because ergothionine, even though it's in very, very low levels, and plants typically have 10 to 100 times less, so 1 to 2 orders of magnitude less than a fungus. But because it's ubiquitous in the food supply as a result of very low doses in plants, it's almost impossible to create a human diet where humans are devoid of it. But you can do that with laboratory animals. And when you make laboratory animals deficient in ergothionine, there's an increase not just in one disease, which is the definition for a vitamin deficiency, but there's an increase in multiple diseases that all relate to oxidative stress. So if we redefined that traditional definition of a vitamin to disease processes, where oxidative stress can cause cardiovascular disease, it can cause neurodegenerative disease, it can cause liver disease and diabetes, then yes, it does fit that definition. But I don't think we're likely to see ergothionine called as technically a vitamin anytime soon. In clinic, however, we need to think about it as a conditional vitamin. There's compensatory mechanisms, so if it's low, other things might be able to help out. But through the lens of looking at the research as a healthcare professional, in this kind of herbal and fungal mushroom space, it clearly comes extremely close to fitting this definition. So Ergo has a broad range of antioxidant and cytoprotective properties. It can act as a direct antioxidant and also an indirect antioxidant. So it can help out in a reaction, or it can increase the expression of other endogenous antioxidants in the cell, as we'll see. It also, it's got a range of kind of targets. It's primarily protecting the intracellular water-soluble protein. So inside a cell, it's a very dynamic place. And many of us now have seen the animations of the machines and motors that drive all of the internal workings of a cell. So the proteins within a cell need to be held in their conformational shape. And if they're not in their conformational shape, just like the gears that work together to move things, if those cogs in those gears get bent out of shape, they can't work anymore. So maintaining the conformational shape of the proteins means that all of that machinery can function properly. So that's its primary job. It's really good at doing that, but it also gets involved in managing lipids and other things as well. There's a bit of controversy about mitochondria, but in 1982, a researcher called Kawano radio-labeled ergothionine and then fed it to animals, and then looked at those animals, and the ergothionine accumulated inside the mitochondria. So that research hasn't been repeated yet, and this is why it's a bit controversial. But it makes sense that this is a very small molecule. I think the big deal is that the transporter proteins haven't clearly been identified on the mitochondrial membrane just yet. But I believe that it is helping out with mitochondria as well. So as I've already said, the ergo is a secondary antioxidant buffer or backup in extreme kinds of conditions. And it's important for us to make note of that. So it's not going to be acting constantly. So it's a secondary backup, and as I said, it's only comes into play when glutathione is depleted and it needs time to regenerate. So we need to be thinking of ergo not as a full-time antioxidant. It's kind of like a part-time antioxidant cytoprotective, and it only comes into play when we need to act out plan B because plan A is not working properly. So as a result of this, it doesn't have much activity at all in healthy tissues. It only gets engaged when there's stress in a particular tissue. And you might be wondering at this point, well, how does it do that? A good question. I will come to that and explain it quite fully for you. So this makes ergothionine an adaptive antioxidant. So it's there, but it only engages when it needs to. And it's also very resistant to auto-oxidation. So some antioxidants, so uric acid in the bloodstream, it performs the role of an antioxidant, but if the levels get too high, it can leave the bloodstream, it can get into the surrounding tissue where under the influence and the presence of oxygen, it can become quite pro-oxidant and inflammatory. Vitamin C undergoes the same processes, so do some of the purified alpha tocopherol, particularly. If you get too much of that, it can not be antioxidant, can be pro-oxidant. So it's important to understand that ergothionine has got this safety profile around it, which is really quite stellar, and it's totally kind of unique for any type of antioxidant. So as well as being the resistance to auto-oxidation, because of this, it doesn't interfere with the healthy activity of reactive oxygen or nitrogen species. So when a cell creates a certain amount of reactive oxygen species, that's a natural consequence of respiration, and the production of those molecules interact with NRF2 to drive the production of all of the antioxidant defenses that keep the cell naturally healthy. So if you have too much antioxidant activity within a cell, you can actually turn off the mechanisms that keep the cell healthy and cause challenges and disease. So here's the point where we can talk about how it actually works, this magical role of being adaptive and only get engaged when it's actually needed. So on the right, ergothionine exists in its thione form, and in this thione form, it is not an antioxidant. So ergothionine can exist in two forms. The other form is a thiol, and it can exist in these two forms based on the pH of the environment that it's in. So the fact that this molecule can be two different shapes, even though it's the same molecule, means it's a tautomer. That's the scientific technical term. So when cellular stress goes up, reactive oxygen species get produced. So that has the effect of making the environment in the cell more acidic, so the pH lowers. So as the pH lowers, and in the scientific papers, they talk about a pH of around 7.4. So when the pH gets below that, it turns into its thiol form, and then it becomes antioxidant, and it can deal with the oxidative stress, bring the pH back up, and then it can flick into its thione form. So as the cells are being stressed, glutathione is engaged with dealing with whatever's going on. If there's excessive stress and all of the glutathione gets depleted in a cell, which can easily happen, let's say, in an endothelial cell in the vascular system, it can easily happen in a hepatocyte in the liver, just as a result of having too much sugar or drinking too much alcohol, then those nutrients get into those cells, they have to be disposed of, and the disposition of those nutrients, the metabolism of them, creates excessive amounts of oxidative stress, which deplete the glutathione. So the cell's got all this stress, the glutathione's low, and then at that same time, the cell pH goes down. So that's when the ergothionine comes back online. It's a very elegant and very illustrative of how clever nature is, and it adds a lot of kind of benefit to the fact that we should probably consider this a vitamin because it so naturally fits into the way that our physiology works. And the expression of the transporters, which influence the concentration of this molecule throughout the body, are expressed in tissues which have got the highest levels of oxidative stress, which is another clear sign of how important ergothionine is to various cells and tissues in the body. I was saying before that it has these indirect applications as well. So it can influence NRF2 signaling. So NRF2, as I was just saying, it's kind of like it's a transcription factor that gets released as a result of stress in the cell. So there's a stressful message. The nuclear factor, the NRF2 receives that message. It gets dislocated from where it's held on the inside of the cell membrane called keep1, translocates into the nucleus where it transcribes on the antioxidant response element. And that's about 250 genes that code for all of our antioxidant defenses. So depending on the message that gets delivered, NRF2 transcribes a particular part of that and produces a particular protein, which will be some antioxidant enzyme or some other factor that's involved in dealing with stress. So that's how it's indirectly antioxidant. So the output of that could be superoxide dismutase or glutathione reductase or catalase, for example. The way that it does that isn't clearly understood yet. And I mentioned the fact that it controls the conformational shape of proteins. So it does that as an antioxidant, but it also encourages, via mechanisms that still aren't clearly understood, it must interact with heat shock factor, which increases the production of heat shock protein 70, which is the main intracellular chaperone that does, if a protein gets bent out of shape, it can repair it and put it back into shape. So for those of you that have been following my herbal work over the years, you'll know that herbs that can have this action typically fit into the category of being adaptogenic. So adaptogenic herbs have got a significant capacity to regulate that aspect of cells, and that's how we can keep a whole body healthy, by making cells resistant to stress in the form of dysregulated proteins, which down-regulate the efficiency of our cells. Ergo's got a really interesting relationship with inflammation, and nuclear factor kappa B is the kind of the master regulator, if you like, of cellular stress and inflammation. And as stress goes up in the cell, nuclear factor kappa B gets expressed, and the expression of that increases the gene activation, which codes for the production of the ergothionine transporter. So as cells are undergoing stress, NF kappa B gets induced as a mechanism to control that. And then at the same time, it's telling the cell to bring more ergothionine in because I've got stress here and I need more support. The glutathione and whatever's not helping out as much. And then as the glutathione increases in the cell, it helps to shut off the activation of the nuclear factor kappa B. So you've got this very elegant integrated system, which, when everything works properly, the cells just won't go wrong and won't get become dysfunctional or develop disease. And we're all adept at using anti-inflammatory herbs in clinic. Things like curcumin and boswellia come to mind. But this new knowledge that ergothionine plays a role in the control and regulation of nuclear factor kappa B clearly puts it on the table as an important anti-inflammatory support agent in those types of protocols that we're using. There's not a lot of data on ergothionine, and what we do have is quite startling, and it tells us that different countries, due to their different lifestyles and dietary patterns, have got quite divergent levels of ergothionine in the diet, and therefore the levels of ergothionine in the population are low. And a modern westernized diet is deficient in ergothionine, and it's best represented by the USA, being the lowest on the list here at just 1.1 milligrams a day. And to be healthy, you need around 4 to 5 milligrams a day of intake. And the reason that Italy is so high up on the list here is probably because Italians love mushrooms, and the porcini, or the Boletus edulis, which is very, very high in ergothionine, is like the national kind of mushroom. It's the signature mushroom, of that country. If you look at mortality rates for neurodegenerative diseases like Alzheimer's and Parkinson's disease, they also correlate with the dietary intake of ergothionine as well. So there's more to say on that, but it's a very interesting picture that emerges, and it's a clear example of the need for the population in general to have more ergothionine consumption. For those of you that want to read the research, I put this slide in here just to show us that the ergothionine transporter has got a couple of different names. So the SLC224A, that's the name of the gene that codes for the production of the protein. So that's the most accurate name. OCTN1 is what's currently used in the science, but it's not very accurate because it isn't a transporter for organic cations. It's primarily a transporter for ergothionine, ergothionene's metabolites, and also selenoneine, which is the same shape, but instead of sulfur, it's got a selenium molecule attached to it. So that's a whole different story. Tuna are very high in selenoneine, for example. And then as you can see on the table here, there's a list of other molecules that get transported, but they're significantly less than the ergothionine. The ergo transporter gets expressed, and this is just the current knowledge. There'll be a longer list as we go through time. There's these 58 different tissues that have been shown to have high levels of transporters, or in the case of red blood cells, which don't have the transporters, but the red blood cells have ergothionine inserted into them during their manufacture in the bone marrow, and red blood cells have got one of the highest levels. But the gastrointestinal tract has got very high levels. The eye, the small intestine, the bladder, sorry, I meant to say the kidney, where it gets absorbed in the small intestine. If it gets into our bloodstream and into our kidney, the kidney's like, no, I'm not excreting you. You're really valuable. I'm going to resorb you, put you back into circulation. So, and many of the conditions that are associated with low levels of ergothionine match up nicely with this tissue distribution here. And those diseases I listed out here. So because it's still early days, and scientists are a cautious lot, so they're not fully saying low ergo is the cause of these diseases. It might be that as we get older, we're less efficient at absorbing ergothionine, for example, or those transporters might not work as well, and that might be the major issue. But whichever way we look at it, whether it's diet, there's polymorphisms that we know about in terms of those transporters, and there are definitely some diseases that involve the polymorphisms. There might be low gastrointestinal uptake. There might be impaired resorption in the kidney, for example. But the ones that I've listed on the slide here, taken from this paper that I'm quoting, and if you put that PMID number into, you can just see behind my right arm there, into PubMed, it'll take you to this free paper. It's a really interesting read. So you don't live as long if you've got lower levels of ergothionine. As you get older, you're more likely to be frail. You're more likely to be cognitively impaired. Across all of the spectrum of those neurodegenerative diseases, you're more likely to have cardiovascular disease, chronic renal disease. The highest correlations, as we'll see, are with macular degeneration. So the eye is a tissue and an organ in the body that has a great need for ergothionine. There's some association with cataract development as well, and preeclampsia. So pregnant women that consume mushrooms have healthier pregnancies and less preeclampsia, less type 2 diabetes. Children that are more normal in their birth weight, so less, below average weight and less above average weight children. But the topic of this presentation is the cognitive health. So we need to just take a closer look at the cognitive stuff. And we can come back at another point in time and look at those other conditions. So ergo exerts its effects via multiple models of neurodegeneration. I just touched on a few things. So there's neuroinflammation, there's or the inhibition of that. There's reduction in stress. That could be through chelation of metal ions. So high levels of iron are quite closely associated with that. It may be the preservation of mitochondrial function, for example. So I've listed out here where the ergo transporter protein is expressed across the cognitive tissue. So it's not just in neurological tissue, but it's also in the vascular tissue and also in the immunological tissue. So the brain microvascular endothelial cells, BMEC would be a quicker way of saying that. So they're the specialized cells that line the interior surface of the blood vessels in the brain, and they form the blood-brain barrier. So these cells are much more sensitive than peripheral endothelial cells to oxidative stress. And ergothionine has antioxidant and anti-apoptotic effects that are well understood from animal models in these particular cells. And ergo helps reduce the negative effects of a long list of known oxidative kind of molecules like oxidized cholesterol, 7-keto cholesterol, for example, which causes all sorts of downstream complications. The choroid plexus is really important. It produces cerebrospinal fluid, which is important for brain health. Neural stem cells are limited to certain regions of the brain, like the hippocampus, for example, and they're essential for neurogenesis, and that's crucial for learning and memory. So, there's a clear association there. In animal studies, ergo has been shown to protect neurons against neurotoxicity, for example, from beta amyloid toxicity. And the whole Alzheimer's thing, which is very, very likely an inflammatory condition in the neurons, which switches the cell into producing that amyloid beta. Amyloid beta is a byproduct of the inflammation. It's not like the cause of the disease. So this is how ergothionine, may have an impact on Alzheimer's disease by organizing the cellular environment to be less inflammatory. The astrocytes and glial cells, they provide structural support. They help to regulate the extracellular environments, and they help to modulate the transmission through the neurons. They contribute to blood-brain barrier activity, and because the glial cells are immune cells, they play a role in inflammatory conditions like certain psychiatric disorders or whatever. So what ergothionene's doing, across all of the tissues in the brain is really quite comprehensive and important. So there's a bunch of animal studies. I'm just going to pick on a couple of them and then look at the human data as much as possible. So when you give healthy mice the human equivalent of about 25 milligrams of ergothionine a day, so the mice got 4 to 5 grams. I did the calculation based on surface area and came up with 25 milligrams for 7 weeks. The P90 is the age at which 90% of the population dies, and the mice that got given the ergo lived 30% longer. So it was 868 days compared to 672 days. So they had a slower, significantly slower age-related decline in their weight and their fat mass and their average and maximum movement velocity. So they were much more active and much less frail. They had a significant suppression of the increases in important plasma biomarkers. So you can look at the plasma and you can see markers for aging, quinolinic acid and kynurenine. There's a whole bunch of things that were improved, and it prevented age-related decline in the hippocampal neurogenesis. But this protein, TDP43, when it aggregates, it's been recently recognized as a new form of dementia. So it's similar in the way that amyloid beta can come together and form clumps. So this particular protein has now precipitated the need to have a new disease category because many people that have got Alzheimer's disease don't have accumulation of amyloid beta, they have accumulation of TDP-43. And the disease is now being called LATE, which stands for limbic predominant age-related TDP-43 encephalopathy. Real mouthful, that's why we're going to call it LATE. And this doesn't typically happen to people until they're aged 80 or above. And researchers suggested about 50% of the population of all Alzheimer's cases are in fact cases of LATE. So the fact that ergothionine in aging mice decreased this protein accumulation is really, really quite significant. Ergo also promoted the microglial shift to the reparative anti-inflammatory M2 phenotype. So this is seen as being extremely beneficial in neuroinflammatory conditions, particularly things like Alzheimer's disease. And beyond just being anti-inflammatory and promoting tissue repair, that M2 phenotype is associated with enhanced neuronal survival. So clearly, really important. So that's it for the animal data, almost. Let's now turn our gaze to the human data, where there is very strong and significant associations. We don't yet have the causation, but this data is incredibly compelling. So this data comes from people that were being looked at in various health institutions in Singapore. So there's a healthy group of people, and on the left-hand side, there was 544 of those. And on the left-hand y-axis, we've got plasma ergothionine levels in nanomolar amounts. And you can see in the healthy category, there's a light blue box, which is the majority of the people. There's little bars at the end of the lines that come out from each end. So that's kind of like the outer limits of where the data was collected, but that box represents the most statistically significant group of those people. And then the line in the middle of the boxes is like the numerical median, so the most common number that was achieved in those assessments. And then at the bottom, we've got the disease state. So we've got that line matches up with about 1200 nanomolar concentration. So with mild cognitive impairment, that number falls just below 1000. With Alzheimer's disease, it's around 800. With vascular dementia, that midline is around 800 or 900. With Parkinson's disease, it's a very similar number. And then with macular degeneration, it might drop down to 750 or a bit less than 800. And macular degeneration had this, the biggest, clearest association. So this data is really quite impressive. Part of the challenge is that we don't yet have a blood test for ergothionine that's commercially available. It's just a research tool at the moment. And we're probably a couple of years away from, maybe 1 or 2 years, according to the researchers, from having access to that. Once we have access to that, I think the whole ergothionine landscape will significantly open up. So on the right-hand side, we can see where the healthy range is and where the low and unhealthy range is. So we can take this data and say every patient should be taking mushrooms every day, or at least 2 or 3 times a week, according to that epidemiology stuff. Or to be safe, take an ergo supplement. Just 5 milligrams a day would be enough across the course of your life. The ergothione's like a lifestyle strategy. You can't do all of your exercise just in 1 week of the month or 1 month of the year or 1 year of your life. You've got to do it regularly and consistently across your whole life. Same with a good diet. You just can't do a good diet 1 month a year and then eat crappy food the rest of the year. So ergothionine fits into that category. We've got to be doing it regularly throughout our lives to have these types of benefits. If you group all of those people together, all of the unhealthy people and all of the healthy people, and look at that same data, this is now what it looks like. So each one of those dots represents health, represents an individual person. And on the healthy side, those blue dots, there were 14 blue dots above that level of 3,500 nanomolar concentration. So the average healthy person was around 1,200, and the average unhealthy person was around 800. So we've got to get that line up above 1,000 across the course of your life to effectively prevent these types of diseases. But all the other ones as well that we're not talking about. Cardiovascular disease has the same associations associated with it, for example. So there's a lot of research looking at these types of associations where they take cohorts of people and then they'll look at the blood. So they might look at the blood and make some correlations, or they might look at the blood 1 year and then wait 5 years, or wait 2 years, or wait 20 years, and then compare and see what's happened. So I haven't done an exhaustive presentation on that. I have that in other locations, but just in the interests of time, I was just going to talk on a couple of them. So this is Japanese research, which was relatively recently conducted, just a couple of years ago. And inside the purple part of the Venn diagram, we've got 33 dementia patients, and there's a list of metabolites which have been shown to be either increased or decreased. And then we've got the kind of the aqua-coloured frailty group, 15 people, and we have a group of people with cognitive impairment. There were 6 of those. And then where the Venn diagrams intersect, ergothionine and tryptophan were the lowest and correlated with all 3 of these conditions. So for people with dementia, frailty, and cognitive impairment, ergothionine and tryptophan are kind of like the peak, and everything's downstream from there. In the overlap between dementia and frailty, trimethyl histidine and S-methyl ergothionine, they're metabolites of ergothionine. So even the metabolites, even though I haven't been talking about it, they have some biological action as well, which is less clearly understood. Low levels of methionine play a role. So you can do this data a number of different ways, and the research pretty much always comes out with ergo being the most highly correlated metabolite that's low in association with the diseases. It's also the same with cardiovascular disease as well. So the researchers are suggesting that, as well as ergothionine, being causal, although we don't have that data yet, it's also predictive. So there's a strong need to have data that can predict the development of future cognitive decline. So in this study, where a bunch of subjects that were 73 years old were followed for 5 years, and then their ergothionine levels were looked at. So the serum concentrations in this study were using the imperial system, so between 20 and 100 micrograms per mil was the typical range that was seen. And patients without dementia, on average, were 31.5, and with dementia, 20.7. So the higher the number, the less dementia you're going to have. And this matched up with not just with plasma ergo levels, but also with neuroimaging of cerebrovascular disease and brain matter atrophy, like white matter hyperintensities, for example. And they also did some tests on cognition and function. So the ergo correlated with all of these things. And the correlations flowed in this particular way. So if we had low plasma levels of ergo, there was an acceleration in the decline of all of the cognitive functional domains that were being looked at. and there was a faster functional deterioration. So people became less physically capable, not just cognitively capable. So they became, I guess in a sense, you could say they became more frail. And the plasma ergo was very, very strongly linked to those two things I was talking about just previously. White matter hyperintensities, which show up on an MRI, and they're to do with the vascular system causing detriments in the white matter of the brain. And they're quite a common hallmark of this. And interestingly, when people consume more than 2 cups of coffee a day for more than 15 years, these white matter hyperintensities also increase. So there are other things that are at play and may be valuable. The coffee thing's a bit controversial, but high doses over long periods of time, just 1 cup of coffee a day is a beneficial thing. And on that previous slide, if you go back and look at it, caffeine is linked with positive associations with dementia if you get the right dose. But plasma ergo is also closely linked with just generalized brain atrophy. So you can measure the global thickness of the brain and the volume of the hippocampus, and the thickness decreases and the hippocampal volume decreases as well. This is another study that came to exactly the same conclusions as the previous study, except that was just conducted over a 2-year period. So patients that had the lower levels of ergothionine developed Alzheimer's at a rate that was 12% higher than the other group. So when you look at mild cognitive impairment, not all patients progress to Alzheimer's disease. Only about 15% of them over the age of 65 progress to Alzheimer's within a 2-year period. So this study is saying that patients with low ergothionine, they're likely to progress to Alzheimer's disease 12% more than what you would consider to be the average. This next study is a combination of a human study and also an animal study. So the data that is expressed on this slide was the first publication, and they didn't publish any of the animal data on this one. On the next slides, we'll see the animal data. So this is one of the few human clinical trials where ergothionine has been given and then cognitive function has been assessed. So people got 5 milligrams of ergo for 12 weeks. They had cognitive function assessed at 0, 4, 8 and 12 weeks. So every month basically. One of the weaknesses of this study is that the people weren't very demented. Their cognitive function, wasn't actually that impaired. So using the mini mental state exam, you need to be below 26 to be considered to have mild cognitive impairment, and the averages in these both groups were approximately 27. So they were kind of borderline. So perhaps this study doesn't have huge value, but the animal bit of the study adds value, which makes this a very, very interesting and extremely kind of insightful take on how ergothionine works. So when at the end of the study, when they looked at psychomotor speed, verbal memory, simple attention and sustained attention, these all improved significantly against placebo, but you wouldn't call the improvements clinically relevant because there was not much kind of cognitive decline present. So you can get statistical relevance, but that still might not be big enough of a difference to actually have a clinical meaning. So the clinical aspect of that wasn't so good. But the researchers were really keen to understand how ergothionine actually was involved in affecting cognitive function. So this is where the animal bit came in. And they were interested in ergothionine's relationship and its influence over a receptor in neurological tissue called TrkB. So TrkB, or the tropomyosin receptor kinase B, sometimes it's just called receptor tyrosine kinase. It's the receptor for brain-derived neurotrophin factor. It's also a receptor for neurotrophin 3,4, which is another neurotrophic molecule that's endogenously produced, although it has less affinity for that receptor. It's also a receptor for some of the lion's mane secondary metabolites. It's also the receptor where psilocybin binds, and it's how psilocybin influences its influence over behaviours and cognitive function. Psilocybin at the 5-HT2A receptor is what causes the psychedelic kind of experience. So TrkB's got a lot of interest in it, and it's not just TrkB, but there's particular pathways that flow on from the activation of this that are also important. And I didn't want to talk about that because it's just a little bit too technical. So when BDNF arrives at this receptor on a neuron, so the receptor sits in the cell membrane, and there's an outside bit and there's an inside bit. So when the BDNF binds to the outside bit, it creates internal changes to that receptor, which allow the message to be sent. So the receptor dimerizes, so you get two of them come together. The internal arrangement gets phosphorylated, so phosphorus molecules get added, and then that regulates the particular signal that's coming down through the BDNF. So if that BDNF signal, if the intent of it is to say, you don't have enough synapses going on here, you know, that synapse, that synapse, that synapse, this one, they've become dysfunctional. We need to repair them. We need to regrow new ones. So that might be the message that gets delivered. And for that message to act out in terms of physically doing the task by the cell, this process of phosphorylation is a crucial step. It's the first step in like the initiation of the message. So if you don't get the first step right, you can't get the survival, you can't get the growth, you can't get the synaptic plasticity, you can't get the cognition enhancement. So this phosphorylation step is really quite important. So this is where the animal data meets the human data, which is what, you know, gets really interesting. So when they gave the ergothionine to the animals, what they observed was that the phosphorylation of TrkB was enhanced as a result of the ergothionine, and that was significantly involved in the enhancement of the cognitive function. So the cognitive function enhancements that you see are as a result of ergothionine intracellularly enhancing the landscape, the internal environment, so that the internal part of that receptor is more capable of delivering its message. So often in science and in these experiments, we get transfixed with the actual messenger. So, you know, we could use the example of Ergothioneine A, which has everybody transfixed at the moment in terms of how valuable lion's mane mycelium is in terms of helping, you know, cognitive function. But there's also hericenones, and we've also got the data that tells us that the very earliest studies in lion's mane were conducted on whole mushroom. And we had that discussion very early on, and the whole lion's mane mushroom is quite high in ergothionine. So I haven't got a problem with Ergothionine A. My contention would be that if you want the very best lion's mane product, and we can't yet do this because Ergothionine A from mycelium is not commercially available, it's just an experimental tool. You would make a product that had the mushroom or the fruiting body with all of the known traditional constituents optimized, and then you would add to that, you know, a mycelial extract to get the benefit. And then you could get all of the synergy that you need to make this function properly. So you could, if you gave all of the Ergothionine A in the world There are human clinical trials now. There's one to show that you can have benefits in Alzheimer's disease. But we've got all of this data on the other side saying if you just give ergothionine, you will prevent Alzheimer's disease and you may be able to treat it. So we haven't yet put the two things together. We have in the very first case when mushroom was being experimented on and showed that it improved cognitive function, it decreased frailty, for example. So this experiment to me is like really important because it tells us that we've got to get an understanding of what's actually going on so that we can be fully cognizant of all of the synergy that's involved. So ergothionine is making the environment of the cell more receptive to the messages which are necessary to improve the situation. And if we're able to naturally increase BDNF, then we might not even need RNA-CNA or the hericinones that do that same type of job. And that's the whole benefit of maintaining health throughout your lifespan. You know, you might not need the secondary metabolite. You might just need the primary metabolite, the ergothionine, to keep you healthy. So that was a long bit of a rant there. But clearly, I think you get how important this particular aspect is. So the next part of this puzzle, which was particularly fascinating, is that the part of the brain that makes these things, so the TrkB receptors, for some reason, and researchers don't know why, those TrkB receptors get packaged and put in little vesicles and released into serum. And you can measure them in the body. Part of the reason is likely to be that TrkB also plays a role with enteric glial cells in the gastrointestinal tract that control immunological and nervous system function. So this might be, part of the communication mechanism or part of the maintenance of the gut-brain axis. No one's really quite sure. But in any case, when they looked in the humans, since they took blood to do this, when they looked at the humans, the amount of phosphorylated TrkB in these vesicles compared to the unphosphorylated TrkB was better. So in the humans that got the ergothionine, the phosphorylated TrkB, which is what you want, was higher, and the unphosphorylated TrkB was lower. And when they crunched the numbers, there was statistical significance looking at that fact, comparing it to all of the domains in the cognitive test. So composite memory, verbal memory, processing speed, visual memory, reaction times, complex attention, cognitive flexibility, executive function, and working memory. There was very significant statistical significance between those domains and the levels of phosphorylated TrkB and unphosphorylated TrkB. So clearly, there's some really important stuff going on here with regard to the way that ergothionine is working. So that was my last slide on the cognitive piece. I just wanted to finish off the discussion by saying that ergo is also associated with these other risk factors, cardiovascular disease, and there was a study by Smith, and went over about 18 years, I think it was, or it might have been 20 years, there was a lower risk of coronary disease, there was a reduced risk of cardiovascular mortality, there was a reduced risk of all-cause mortality, and your chances of those things happening were between 15 and 20% less if you had higher levels of ergothionine. And it matched up with what the researchers described as a health-conscious food pattern. I mentioned this earlier, that ergothionine can help in type 2 diabetes. So when you give people 100 grams of agaricus bisporus, so button mushrooms every day, which have got 3.2 milligrams of ergothionine, the scientists measured it for 16 weeks. There was a reduction in oxidative stress. There's other things in the mushroom which may have played a role as well. And then in a relatively recently published trial that was really well conducted, at 20 milligrams a day, ergothionine improves sleep in elderly people, and didn't just make them sleep better, but when we get older, we have reduced quality of various phases in our deep sleep, and we need deep sleep to regenerate our brain, turn on our lymphatic system to get the cleansing and remove all of the waste from the day's metabolic activity. So for aging people, this may be a clue into one of the kind of corollary mechanisms whereby ergothionine improves sleep, improves deep sleep, improves the lymphatic system, which therefore reduces the chances of cognitive decline and the development of Alzheimer's, for example. So let's look at the safety, what's going on here. So I've been saying all along that ergothionine is safe, and there's the scientific kind of understanding of the way the molecule works. But in animals, you need to give them 800 milligrams per kilogram a day in rats, and that equates to about 9 grams to a human dose. So that level of safety is enormous. So that 9,000 milligrams of ergothionine, and so it's not ever going to be possible for anybody to take too much. There's been no adverse effects reported with any human intake, and in the US and in Europe, synthetic but nature identical ergothionine. So you can take the genes out of a fungi and you can put them into E. coli or a different type of fungi, a yeast, and the cells go to work as little ergothionine factories and make L-ergothionine, which is identical to the fungal version of it. So that's approved for use in the US and Europe. And in Europe, it's approved for infants, children, adults, and it's in pregnant women, it's approved, and also in breastfeeding women, but it's only approved if they get a source from a naturally growing mushroom or plant. Mushrooms are the primary source. So it's got GRAS status in the US. That happened in 2018 at a dose of 5 milligrams a day. But I think, clinically, 5 milligrams a day would be kind of like the basement dose. In Australia, where I'm recording this, it's only approved for topical use at the moment. It plays a very important role in protecting skin against oxidative stress. So mushrooms are a producer of ergothionine, so it's the best source to get them from. So the emerging data strongly suggests that adults and children can have 5 milligrams a day approximately as a maintenance dose. For a treatment dose, adults up to 30 milligrams a day. Treatment doses in children a bit lower, maybe 20 milligrams a day. So there are going to be situations in children, particularly children that are more physiologically vulnerable to stress, and children that have got neurological challenges, the early stages of autism, for example, where children might be having a response to low levels of glutathione, so the nervous system can't respond to the stress well. That's possibly a really important place to use it. But there's a lot of situations, where ergothionine is going to play a role with oxidatively stressful situations. And in pregnant and breastfeeding women, 5 to 30 milligrams per day. And in terms of breastfeeding, a very interesting little snippet of data is that in women that are breastfeeding, the mammary epithelial tissue that produce the milk express 4 times more ergothionine transporters than non-lactating breast epithelial tissue. So clearly there's an evolutionary imperative for the mother to provide the child, the developing child, with levels of ergothionine. And there's some compelling evidence from animal studies that ergothionine is important for proper neurological development and neurogenesis. And it's not just humans that we're talking about here. All vertebrates have got ergothionine transporters, so all of your pets need ergothionine as well. So, your cats and dogs, for example. So mushrooms are the best source. You can't really rely on vegetables, even though some plants like red beans, for example, oats, contain some levels. It's about 1 to 2 orders of magnitude less. But we now understand that the practices of growing modern vegetables and crops on a large scale damages the mycological environment. So the soil fungi aren't as healthy. So there's not as much transfer from soil fungi into the plants. So modern agriculture is producing vegetables which have got low levels of naturally occurring ergothionine. I mentioned the porcini earlier as being the highest, but the Pleurotus citrinopolitus, I think I pronounced that right, the yellow oyster is the best place to go in terms of a freely available mushroom that's cost effective. And I've quoted 3.9 milligrams per gram dry here just from one of the research papers, but I know from my first-hand experience that the different varieties of this yellow oyster can have vastly different levels. And the one that's being used commercially can be up to 10 milligrams per gram of dry mushroom, which makes it a real ergothionine powerhouse. But if you're eating mushrooms on a regular basis, the ones that are considered high are 1 milligram or above. So maitake lion's mane, pearl oyster and king oyster, all very delicious mushrooms. So when you eat those, you're getting significant amounts of ergothionine. And then for mushrooms that are considered to be low or moderate, they're below 1. So shiitake is on that cusp of 1 milligram per gram dry. The ratio Organoderma is a bit lower. And then the agaricus mushrooms, the crimini white button and portobello, they're about half a gram at the most, half a milligram per gram. So they're still a relevant source, and if you eat them a couple of times a week, you can get enough to maintain your health. And tempeh is also a good source. It doesn't have very high levels, but when you eat a meal of tempeh, you're eating a couple of ounces, and that increases the number. So, for the people that were, originally consuming tempeh as part of their traditional diet, that might have been a clever and an unrecognized way of getting the right levels of dietary ergothionine. So this data is just a repeat of what I said, very, very early on in the presentation. So I won't, go over all of the details again, except to say that when we're thinking about consuming mushrooms, one fresh portion a week, which equates to about 12 grams of fresh mushroom, which is about 1.1 grams of dry mushroom, gives measurable benefits in improving cognitive function. When you get above 2 serves, it can decrease the risk of developing mild cognitive impairment, and at higher levels, decrease the risk of neurocognitive diseases. So on the graph that you can see here, there's an inverse relationship. The more you eat, you continue to decrease your odds. And when you get out to the end here, which is 9 serves a week, your chance of your odds ratio is about 0.25 approximately. If I can line that up, maybe it's 0.2. So you've got an 80% reduced risk of having cognitive impairment. So if you can eat mushrooms every day, do it. If you can take a mushroom supplement, so if you took a mushroom supplement, let's just say you took a lion's mane that was a whole mushroom preparation at 1 gram a day, that would be the equivalent of very close to 1 serve. And if you did it every day, you'd have 7 serves. So your chance of developing cognitive impairment, is reduced by about 75% if you did it across the course of your life. So why wouldn't you do that? It's impossible in my experience to eat mushrooms every day. There are mycophiles. I follow some people on social media that eat mushrooms every day, but I love mushrooms. I'm a practicing herbalist. I practice what I preach, and the only way I can get mushrooms every day is if I take a supplement. So I have them as teas, and I take them as capsules, and I eat them as often as possible. So that's my advice to you as well. And here is that advice, written down in as much clarity as I can. So there's no right and wrong answers here. The right answer is just eat mushrooms regularly, start and never stop. Just like exercise, start exercising, never stop. Start having a good diet, never stop. Start, thinking good thoughts every day, never stop. There's all these lifestyle things. Sometimes they're easy to say, sometimes they're hard to do. So with mushroom consumption, if we want to prevent neurocognitive decline, which is the new DSM category for what we used to call mild cognitive impairment, you need to be consuming mushrooms twice a week, according to that epidemiological data. Or you can have a whole mushroom extract, as I was just describing, and I'd recommend around 1 to 2 grams a day, probably 1 gram a day if you're going to commit to it across the course of your life every day. That would be enough. And as insurance, I would add 5 milligrams of ergothioneine. Now, if you're doing 5 milligrams or 10 milligrams of ergothioneine from golden oyster, if that golden oyster is in fact a 1-in-1 extract, then you may not need to do that 1-in-1 extract. But if you're concerned about your cognitive health, I'd do the golden oyster as well as lion's mane, because lion's mane's got the best track record. And then if you get sick of lion's mane, you can switch it out with shiitake, or you can switch it out with maitake. Doesn't matter what whole mushroom you're having, because it's to do with the ergothionine. And then when you have mild neurocognitive decline, so this is where there's natural decay in our cognitive function as we get older, and from about 40, it can be 0.2 to 0.5% decline in neurons, throughout time. So at a certain point, we can start to notice it. I'm 65 next month, and I'm still bright and cognitively alert, etc. But I make more little annoying mistakes than I used to, for example. So that's where you can notice it. So once you start to dip below that normal line, and you can measure it on, say, the mini mental state exam, then you need to up your dose of fresh mushrooms. You need to up your dose of the mushroom extracts. And my perspective is that give up on all of the other mushroom extracts and make sure at least that you're taking lion's mane. By all means, you can take the other mushrooms because they have other benefits, but lion's mane would become a staple. And if you want to match the clinical trial data in this category, 3 grams a day will do that. And I would add 15 to 20 milligrams of ergothionine. And it's possible to bring your cognitive function, back towards normal if it hasn't suffered too much. I wouldn't do this in isolation of herbs. Standardized ginkgo extract with ginkgolides and bilobelide, it's got good research. Saffron at the right dose, bacopa, bioavailable curcumin. I'm just having the senior moment. The rosmarinic acid from the mint family, so spearmint and lemon balm and sage, etc., rosemary, they're all beneficial as well. Resveratrol, green tea, omega-3 fatty acids, etc. But this is the way to go in terms of mushrooms. And then when it's moderate to severe, we need to increase the dose even further. So have mushrooms more often during the week, increase the dose of the mushroom extract, maybe up to 5 grams a day of the lion's mane, and increase the dose of the ergothionine. All of those herbs that I just mentioned on that previous slide, they're all relevant as well. If you can get access to a quality liquid culture mycelial produced erinacine A, where you can get 5 milligrams a day, which is what the research is, then you could add that to this protocol as well. And I think down the track, we will see that. And then there's a long list of beneficial foods that you would add as well, which could be the subject of another conversation. So I hope you enjoyed that first installment of my on-demand webinar series. I will come back to the ergothionine topic again at some point and look at some of the other conditions where ergothionine is valuable. But we will be cycling through, many different categories. We might focus on individual mushrooms, we might focus on individual conditions. So I could do a talk on the benefits of reishi, but I could also talk about, say, cardiovascular disease, and then bring in the reishi, and a bunch of the other mushrooms that might control lipids, for example. So if you've got any ideas on topics that you'd like me to talk about, by all means, submit them. So thanks for your interest. I bet you're busting to ask me questions, and that is the downside to an on-demand format, that you just can't ask those in person. So I suggest that you sign up to the Real Mushrooms Practitioner Facebook page, and then you can ask all the questions you like. And there's a team of us that monitor that page. And then if you want to direct those, particularly at me, you can make a note to say, can I please ask Lee this question about ergothionine from his on-demand webinar, such and such. So thanks again, and I'll look forward to seeing you at the next one.